so here's where we left off, and we may have introduced it, I don't remember, it was 5 days ago, so I think we left off around here somewhere. What we're picking up, to put it in perspective, is what happens at the target cell when a hydrophilic messenger is present. I think we're okay to acknowledge that, well, the target cell must have a membrane-bound receptor, right? We're okay with that? The receptor for hydrophilic messengers in the membrane? However, we said, well, the complexity of our membrane-bound receptors is very much diverse. Okay, so we said somewhere around 20 some odd receptor families. Okay. And so if we're trying to simplify, well, how would these nearly 20 some odd families of receptors, what do they do? Well, we could say probably they're going to open or close an ion channel, or probably they're going to activate an enzyme pathway. Okay. So if we're trying to simplify it, open or close an ion channel, or activate an enzyme pathway. If we look at the different types of receptors, they're probably, as I noted, going to be channel linked, or in lab we use chemically gated ion channels. We use the term chemically gated ion channels, where we just have this really direct type of permeability effect at the target cell. Some receptors may just activate an enzyme, and some may utilize something called a G protein receptor. We'll clarify the G protein coming up very quickly, which themselves may activate more enzymes or open ion channels. So a bit more complex than just transcription. Not that transcription is easy, right? But we're seeing a diverse amount of responses. And so some examples of enzymatic pathways that may be activated in a given target cell. You've probably heard of the CAMP pathway, the cyclic AMP pathway, a similar one called cyclic GMP, or another one called the PIP2 calcium pathway. And so we're just going to introduce them and elaborate on an example called the cyclic AMP pathway. So I already kind of summarized this. In the target cell, what's going to happen when we use these different types of receptors? One, we're going to change the ion permeability at the target cell, or two, we're going to activate some type of enzymatic pathway by phosphorylating. What does that mean? Adding a phosphate, giving it energy, changing its structure, changing its function, making our enzymes more active. So we activate enzymes, phosphorylate enzymes, inside of the target cell. What I want to now do is use some images from your textbook to refer to the diversity of these membrane receptors at our target tissues. The first one, I'm not trying to undermine anything, but I use this term simplistic so you don't feel like you're completely lost or whatever. These receptors that we're going to identify here first are more simplistic in comparison to our second messenger type of receptors. So what I'd like to turn your attention to is figure 5.13, where a receptor is shown in some target cell. What color is the receptor? Pink. The pink purple thing, right? We can see that it's an integral membrane protein. You may notice a kind of V-shaped cutout looking thing. That's the receptor binding site for the hydrophilic messenger. I want you to notice as well, the receptor is more than just a binding site. It's more complex. It's not only the receptor, but it's also what? A channel. And so when the messenger is absent, the channel in this case is closed. What do you think we're going to channel across the target?
Sodium, potassium, and chloride.
Sodium or potassium or chloride or calcium, right? You pick your favorite ion. So we're moving ions. This is an example of a chemically gated ion channel that we introduced in lab last week. An ion channel will open or close when the chemical stimulus is present. Here we're just clarifying that chemical stimulus. We're saying, well, that chemical stimulus must be hydrophilic, water soluble. And so in this case, in the second image, you can see the hydrophilic messenger shown in the purple triangle binds to the receptor at its binding site, changes the receptor's shape, changes its function, and now the target cell has a way for ions to diffuse across the membrane. Just like what we've seen before, even though this is showing an example, it would be ion specific. It would be specifically for sodium, or specifically for potassium, or whichever. And it's not going to let all of those through willy-nilly. It's going to regulate, just like we saw in lab last week, introducing the nervous system. Does that make a little sense? I'll give you a real example. When we leave the second unit and go into the third unit, we'll see how the nervous system controls your skeletal muscles. The nervous system controls your skeletal muscles by releasing a hydrophilic neurotransmitter called acetylcholine. So let's pretend the purple thing's acetylcholine. It's from your nervous system. It binds to receptors, which are chemically gated sodium channels. So what do you think happens? So let's say this is your skeletal muscle. What do you think happens when acetylcholine binds to the specific receptors at your muscle cells, if it's a chemically gated sodium channel? What would happen at the muscle cell, the target cell in this case? If this is a chemically gated sodium channel. It will open and sodium's going to diffuse through. In which direction do you think sodium would flow through a gated channel in a skeletal muscle? Into the cell, good. And it would flow very much like, just like at the nervous system, into the cell, causing the cell to depolarize. To change its permeability, it would depolarize. So some hydrophilic messengers may cause the target cell to change its permeability and membrane potential by opening or closing specific gated channels. Does that make a little bit of sense? Hydrophilic messenger can cause ion flux to change directly. That's one thing a hydrophilic messenger could do. So the takeaway is the receptor is also the what?
Channel.
The receptor is the channel, and it's gated in the presence or absence of the messenger. Some can be very direct like this. Some ion channels not only change the membrane permeability, but if we see activation of calcium channels, as illustrated in figure 5.14, calcium not only changes voltage across the cell membrane, but itself is a second messenger. Calcium is, up till now, you probably thought about, well, I've introduced calcium and it's important when we talk about the smooth ER, right? How can we pumped into the smooth ER and out in the cytosol? So it's an important messenger for like our skeletal muscles to tell them when to contract. Our smooth muscles tell them when to contract. Same thing with the heart. When calcium floods into a nerve cell, the nerve cell will release its neurotransmitters. Okay, so if the ion channel is a calcium channel, not only do we let calcium across into the cell, but then something else is going to happen. So not only are we changing voltage, but the cell may, if it's a muscle, it might contract. If it's a nerve cell, it may secrete, right? It's a messenger, right? So it brings on a whole other realm of possibility beyond just changing the voltage within the cell, like sodium or potassium might do. So adding some complexity. Okay, but it's still a chemically gated channel. Conversely, not all of our hydrophilic messenger receptors are gated channels. Some of them may have an enzyme associated with them on the cytosolic side of the target. So we're still here looking at the target tissue. We can still see a receptor. You may note that the structure is quite different than previously. still have to have a binding site for the messenger. And then on the cytosolic side, there is enzymatic portion. So now this one's a receptor and an enzyme. The enzyme becomes active when the what? When the messenger binds to the binding site. So when the messenger is present, then we activate this enzymatic pathway. These should look a little bit familiar to chapter 2 when we introduced the role of membrane proteins. We said the receptors, the channels, they could be enzymes. Here we're seeing the enzyme activated when the messenger is present. And then when we activate enzymes, we just set some type of reaction in motion. Are we doing okay? Channels, enzymes. They may also be associated with what's called a G protein. The receptor for hydrophilic messengers may also be associated with G protein. They get the name G because they bind to guanosine, one of our nucleotides. So the receptor not only binds to the messenger, but it also binds to this guanine protein. And the G proteins themselves do some very similar things. Activate ion channels or activate additional enzymes. And so initially it kind of sounds the same. But what's different here when using a G protein is the response in the target cell is amplified. When we use our G proteins, the response in the target cell is amplified. What does that mean? Amplified. Like larger. What else was added? Like the ions that are going to do this going to be even more? Yes, even more. Bigger, big time response. Within the target cell, if we activate a G protein in a second messenger system. Okay, so we're seeing these amplifications. And after looking at an image, I'll come back and clarify how the amplification actually happens. So if we look at an image, figure 516, how a G protein can activate an ion channel. So still we're in a target. We see the messenger, the hydrophilic messenger. We see the receptor. We notice the receptor is not a channel. We notice it itself doesn't have any enzymatic activity. What is new is this blue thing. What is the blue thing? The G protein. Okay? It binds to and GDP, guanine diphosphate, and even guanine triphosphate. That looks a lot like what?
ATP.
ADP. So it's also an energy transferring molecule. Its thermal activity is a little bit less, but it's still the same function: phosphorylate, dephosphorylate, transfer energy, take out energy. So our G proteins are complex. How many subunits does it have? 3 subunits. And depending on the pathway, 1 or 2 parts are mobile. And so the image on the right is an activated pathway. So when the messenger is present, the receptor, so the messenger binds the receptor, in that presence that activates the G protein. The G protein, part of it becomes mobile and diffuses across. And look at this, oh sorry.
Yeah, does it become mobile like when a phosphate is added to like GDP or
Yeah, so when does it become mobile? What happens is the receptor changes shape when the messenger is present. That causes the G protein to change shape. When it changes shape, guanine diphosphate is actually let out and guanine triphosphate is let in. So there's no real phosphorylation going on. It's actually just letting this thing out and picking this thing in, which happens to have more potential energy in it. So that physical transfer mobilizes. To answer your question, when guanine triphosphate is added, then it becomes mobilized. And so it's GTP bound, it's activated, then, in this case, we can see opening an ion channel. So you can go on and activate different ion channels. But the ion channels are indirectly affected by the presence of the original messenger. And then we can open the ion channel. So it's going to take a little bit longer to set that motion. Not much longer, it's just still pretty darn fast.
What happens to the other parts of the G protein? Would they disappear?
They didn't disappear. They would still be here. And so just, in this case, the alpha subunit is mobilized. In other pathways, the beta gamma subunit is mobilized. So I mean, that's even adding to another range of variability within the target cell. So it just depends on the cell type, the receptor type, what that cell's designed to do.
So how is it mobilized? Like it looks like it has to come back this way and around or something? Like how does it get?
It would be referred to as a glycoprotein, or not a glycoprotein, a lipoprotein. You see the little lipid portion? So this is still, it's a membrane-bound protein. And so the lipid part makes it soluble. So it can dissolve, float in the membrane just like a phospholipid could. So when it dissociates from the G protein, it's now just diffusing, if you will. It's just because a part of it is lipid soluble. Did I answer your question by chance?
It just looks like it's stuck in there is what I'm trying to say.
Well, it's just a snapshot. It's not stuck. It's gonna physically diffuse over, activate the little, in this case, the channel. And then as soon as the GTP is dissociated, it'll diffuse back towards the G protein. So it's almost like a spring load, like boom, boom, kind of association for simplistic view. And I've got an oversimplified animation I'll show you in just a minute that might help a little bit. But it's sometimes described as like a knee-jerk reaction. Okay, so the G protein, so the messenger binds to the receptor, and the receptor kicks into motion the G protein, and the G protein does whatever it does, and then it comes back. So it's just, they're usually physically even more closer, but they had to separate this out.
The
So.
animation probably will help.
I think it will. But even though we're using a G protein, we're still opening an ion channel. So the target cell, in response to a hydrophilic messenger, may just simply change its polarity by flexing different ions. If it's regulated. The rest of the G protein pathways that we're going to see include the use of our second messenger systems. These are summarized for you in table 5.3. Some various types of second messengers, as noted a moment ago, include the CAMP or the cyclic AMP pathway, the cyclic guanosine monophosphate pathway, CGMP, the diacylglycerol and inositol triphosphate were summarized earlier as the PIP2 pathway. And then also mentioned earlier, calcium can act as a second messenger. So before we dive into anything, let's just think about the word second messenger. What does that imply?
Secondary, not the initial.
It's secondary, it's not the initial message. What is the initial message in a secondary messenger pathway? What would be the
Stimulus? The threshold stimulus?
What would be our stimulus? What would be our, if there's a secondary messenger, what would our primary messenger be? This doesn't quite show a secondary messenger system, but what is the primary messenger? The chemical, whatever it is, illustrated by point 1. Our hydrophilic messenger, your amine, your peptide, your protein, your neurotransmitter, your hormone, your paracrine, your autocrine, whatever, can bind to its receptor. And the receptor is gonna activate a G protein, and then all kinds of other good stuff are gonna happen. And we're gonna produce a second messenger inside of the cell, like influxing calcium, for example. So I want to show an example of a second messenger system using cyclic AMP. Instead of going through your textbook goes in the cyclic GMP pathway, and I think even the FIP2 calcium pathway. But I'm gonna in class just go through the cyclic AMP pathway. Without even studying anything, you can probably begin to see that second messenger pathways are complex. Can you maybe take that away by looking at the picture? Yes, they are. And not shown here is that amplification that I mentioned earlier. Remember I said there's a bigger response? And so the sheer number of molecules are not represented all in this image. So they're trying to keep it simple, saying this and this and this, but actually lots of different messengers will be activated inside of the cell. And so if we look at the cyclic AMP pathway, you can call it the CAMP, but I would caution yourself to, if you do that lowercase to c, cyclic AMP pathway, and it is a second messenger. If you study it though, what should be familiar is there has to first be an initial stimulus. That stimulus has which type of solubility? Which one? Hydrophilic. We're still talking about hydrophilic messengers. They bind to a receptor at the target cell, which is located at the membrane. Well, we see that the receptor is not a channel. When we look more, this one's actually associated with a G protein, which tells us that G protein is going to go on and do something else. It's going to act as an intermediate. The G protein itself is not the second messenger. It's still just functioning with the receptor. If we look at the cyclic AMP pathway, the G protein activates a what? Adenylate cyclase. What is that? An enzyme. In the cyclic AMP pathway, when the messenger is present, the receptor activates the G protein, which can go on and activate an enzyme called what? Adenylate cyclase. Excuse me. Adenylate cyclase uses ATP to make for is called cyclic AMP or CAMP, which is the what? The second messenger. So cyclic AMP is produced after the G protein turns on adenylate cyclase. Second messenger is cyclic AMP. The first messenger is the chemical messenger, like say, you heard epinephrine? It activates a cyclic AMP pathway. The cyclic AMP is not the end result. The cyclic AMP is the second messenger. So the question is, what does it do? It itself turns on different types of enzymes, which are called kinases. Did we use this word before? Some yeses and nos. Kinases. Phosphorylate. These are enzymes that phosphorylate proteins. So cyclic AMP turns on enzymes that phosphorylate proteins. We phosphorylate different proteins, maybe, or what did you say? Add energy, remove the energy, whatever. We phosphorylate something, we give it energy, right? To do some type of work. So our messenger tells the cell to go to, okay, not there, where? Our messenger essentially tells the cell, the target cell, to go to work by activating various protein enzyme kinases, phosphorylating different types of proteins inside of the cell. We're being generic because we have trillions of cells in our body. You want to do this pathway in a trillion times? You're hating it once, right? if we activate proteins, they are going to go to work on a big scale. If this is epinephrine at my heart, the heart will what? Beat faster and stronger. If this is, I'm trying to think of another example. My examples are fading away from me right now. But the takeaway is the amplification happens here, right? A big time response within the cell, and it's variable. A cell may divide, copy a gene a lot, contract hard, contract fast, so on and so forth, by simply activating a second messenger system inside of it. Another classic example of using the cyclic AMP pathway is still epinephrine at your liver. What happens to epinephrine at your liver? Well, we do activate the second messenger system, and what is the net effect? Convert glycogen to glucose. And so we've got to get enzymes to break those glycosidic bonds. Not just one. So one epinephrine on a liver cell doesn't cause just one little glucose to be released. How many glycosidic bonds can be activated in the presence of one epinephrine messenger? 2, is that a lot? 2 sugar molecules, is that a lot?
2.5.
Thousands or hundreds of thousands or millions or even billions for that second. So you're looking at millions, hundreds of millions or more, right? Billions molecules.
From one?
From one first messenger. One epinephrine can cause, say, your liver cell to release not one glucose molecule, but a billion molecules of glucose. So the advantage of the second messenger system is what?
More efficient?
It's a lot more. It's efficient in that I don't have to release a billion molecules for every liver cell. I don't have to release a billion molecules of epinephrine. I just had to release how much? One. So the effect, right, although kind of complicating when you first study it, the payload is for the cell, the target cell.
So how does it know, like, the set point if it keeps releasing? Like, how does it know when it's at its set point?
For
Just the equilibrium.
For epinephrine or for what?
Just for, like, millions for one epinephrine.
So what happens is, instead of just one cyclic AMP being produced, we'll produce, right, I think it's 2, and then each of those can activate a set number of kinases, and those activate a set number of proteins. So we begin to see the amplification. So as long as those intermediates are produced, the pathways are activated. If something's turned on, it's going to do its work. If it's turned off, it won't do its work. And so it's just a matter of pushing the first domino down, if you will. And once the cascade happens, we have a mechanism to set everything back. But question is, how is it maintaining set point? You could just generically say there's a receptor, control center, and effector for everything.
So, like, I guess what I'm asking is, when your messenger initiates it, how does the cell know when to stop if the one messenger?
As soon as it starts, that messenger has to be evicted, if you will. It's going to be either catabolized or just dissociate quickly. And that's going to stop activating the G protein, which would stop activating adenylate cyclase, which would stop making cyclic AMP, which would stop doing this. And those two are just quick reactions. They'll do their work, and then they're done.
So as long as it's in the receptors, it happens. As soon as it leaves, it stops.
The pathway is stopped. Okay. But as soon as the messenger is ejected, the pathway hasn't. We've got all this extra work going on. Well, we won't promote it anymore. We'll let it set in motion, do its work, and then now the receptor could receive a new molecule of epinephrine or whatever messenger you're looking at. And so everything's right. You're looking at microseconds type of on-off pathways. in your textbook, figure 517, you can just kind of visualizing what happens this, then that. This is an animation I wanted to show. This animation is super simplistic, not structurally accurate at all, but I think you can begin to appreciate the cascading effect and the amplified effect within the target cell with this. All right, so this is some generic looking cell, some weird looking receptors, but I think a little better because it shows that G protein in action. All right, so our receptor in this case for our hydrophilic messenger is still membrane bound. The membrane is golden. The effector enzyme in this case, adenylate cyclase, is the blue sphere. And we're going to look at what happens inside of the cytoplasm. The first messenger, so if we want to go to our pretend scenario of epinephrine on a liver cell, for example, that first messenger binds to the receptor. That has to happen first. The G protein is this kind of coily looking thing in this animation. Not structurally correct, but the concept is good. And now when the receptor binds, what's going to happen to that G protein?
It's gonna activate.
It's going to activate. It actually does kind of kick a whack. Boom. And that causes the effector enzyme to produce, in the cyclic AMP pathway, it would be producing CAMP, cyclic AMP. So here we can see our second messenger, cyclic AMP, being produced. Here you can begin to see the amplification. One messenger, or initial messenger, how many second messengers in this pathway? 6. Each of those second messengers can go on, and if we're looking at the cyclic AMP pathway, would activate various protein kinases, which would be in the yellow things here. And now we change the protein kinases. They can go on and have some other protein to go to work. And we can see some output here. Begin to see some amplification. And then those can go on, and maybe if they're looking at different pathways, they can go on and amplify. Whatever. And so maybe those could be the enzymes breaking the glycosidic bonds in glycogen. And the little purple things would be the glycogen molecules. But the takeaway is, hopefully for you, although it's complex at face value, right? It's confusing. But the payload for the cell is that increased response. Does that help a little bit? Any questions after seeing this animation? All right. And if you refer to figure 519 in your textbook, trying to quantitate a little bit the amplification. I should have increased the font from your textbook. I apologize for not doing so. What we're seeing is one first messenger, in this case 10 G proteins activated, and each themselves activate adenylate cyclase, which themselves can produce upwards of 5,000 cyclic AMP messengers, which would directly activate that many protein kinases, which would go on and activate 2.5 million total enzymes in this generic looking. So 2.5 million or a billion or a trillion or whatever is significantly greater than 1. So the payload is an increased response within the target cell. It's very much ordered. In the cyclic AMP pathway though, the G protein definitely always activates adenylate cyclase. So adenylate cyclase is referred to as the effector enzyme. The effector enzyme in this pathway is called adenylate cyclase. It'll be different in a different pathway. And the second messenger is what? It's named after it. Cyclic AMP is the second messenger. And they tend to activate various types of kinases, protein kinases. And what protein they activate depends upon the target. You're looking at a liver cell, you're looking at a muscle cell, nerve cell, whatever. Here's a snapshot of some examples of epinephrine activating a cyclic AMP pathway. In the liver, we identified glycogenolysis could happen, releasing the glucose. Another process called gluconeogenesis, which we use the term, but it's another way to make what? Glucose. So epinephrine, cyclic AMP, cause the liver to not only glycogenolysis, but gluconeogenesis. The same messenger, the same receptor in smooth muscle may cause some smooth muscle to relax, or it may cause other smooth muscle to actually contract. The difference is what? Why the diversity there? Depending on the muscle, wherever it is. A smooth muscle in a blood vessel has a different job than smooth muscle lining your respiratory passageway, right? So what do you think epinephrine would do to the smooth muscle around your mainstem bronchi? What do you think those muscles would do? They would dilate, they would relax, causing your respiratory passageway to be what? Open. Epinephrine, you're probably excited, so you need more oxygen, air, oxygen in. But your smooth muscle, your blood vessels, your kidneys probably ought to, should they relax? Increase filtration? They should constrict. And so same type of tissue, different location, same messenger, opposite results. Because the kinases in the pathway are different in those different cells. And our adipose, our fat tissue, lipolysis. So think about it, epinephrine. We're probably doing something active, so you need what? Not only air, but you need, if I'm running, what do I need? Chapter 3, energy. So I can have some lipolysis, right, breaking down the lipids. And in the heart, right, heart will beat faster, stronger, and skeletal muscles stronger. So the takeaway is we can have one type of messenger, one type of receptor activated the same, but inside the different cell types, the enzymes are different. And so the responses are different. But all what? Key to these we said is what?
Energy.
Enzyme. Protein.
Yeah,
protein
kinases.
So when we activate these protein kinases, the response is what? Amplified. So lots of glycogenolysis, lots of gluconeogenesis, good strong relaxation, good strong contraction, right? Etc. From how many messengers? One. Using the same type of second messenger pathway, but the response in them is different because they're different cell types. Does that help a little bit?
So everything's the same except when it gets to the protein kinase, those are different, which tells it to do something different.
Exactly right. The receptor is the same, the G protein is the same, the effector enzyme, adenylate cyclase is the same, second messenger cyclic AMP. Protein kinase is a generic word. It just tells you it's going to phosphorylate. What it phosphorylates depends on the cell type. Good observation. Additional questions or observations? Table 5.4 gives an example of different messengers, functional classification of their hormone, neurotransmitter, paracrine type of messenger, and then the chemical classification, and then what they would do. So like epinephrine, we know, again, an example of a G protein, right, cyclic AMP pathway. Let's see what choline got on there. Estrogen, steroid hormone, probably causes what? So, check this out. The steroid hormone causes. Steroid hormones cause.
Transcription.
Thank you. Transcription. So we go through estrogen, transcription of the mRNA. Good. Okay. So we didn't talk about the ecdysoids, the amino acids. We don't know. Is it a channel? Is it a second messenger or whatever? All right. So the point of table 5.4 is to show you some real examples instead of just saying a peptide or whatever the case may be. I'm not going to go into this pathway. I just want to illustrate for you that the cyclic AMP is not the only second messenger system. They can certainly get more complex. Another one's called the PIP2 pathway, but what would look common is still we have a initial messenger which has which type of solubility? Hydrophilic, binds to its receptor that activates a protein that activates this enzyme, it all is called phospholipase, which produces for us the effector enzyme makes the second messenger. So here we've got something called PIP2 and the deoxyglycerol, which may go on and activate protein kinases or other enzymes, or maybe even cause the release of, say, calcium, which acts as its own secondary messenger. So it can get extremely complex when you're trying to study a second messenger system. But it's good because the response is amplified in the target tissue, and it limits the number of messengers that our body has from a few dozen, right? It limits it to just a few dozen instead of having trillions of messengers in our cell. Can you imagine how much DNA we would have just to code for that many more messengers? And we would have giant bigger proteins. So is cyclic AMP the only second messenger system? Calcium was highlighted as being a second messenger. I tried to verbally list some earlier. What calcium can do, as mentioned, it may cause a cell to secrete something. It may cause potential changes. What else? Muscle contraction, activating white blood cells, muscle relaxation. There's a question about what happens to the second messenger. We can activate an enzyme to turn it off, degradation, cyclic AMP. We can keep going on for 3 days about what calcium can do. So it's so important for your body, so much more than just bone structure. So let's go back and look at your guided notes real quick. The cyclic AMP second messenger system. What is the effector enzyme? Adenylate cyclase. Very good. The effector enzyme in the cyclic AMP pathway is adenylate cyclase. The second messenger we identified is cyclic adenosine monophosphate. Or you can take CAMP, CAMP. These activate various protein kinases. And the protein is implied when you say kinase. The target cell effects. How can we summarize that? What's the easiest way to say? Do it. Well, what's the apopathic is a way to describe a chemical property. So what about the effects? When we activate protein kinases in any target cell, what's the effect?
Phosphorylation.
Amplified production, various work. It's a result of the phosphorylation that puts this work in motion, and it's variable. A cell may secrete, it may divide, it may whatever, it's so variable. In response to a single messenger which activated the cyclic AMP pathway. The response is amplified, yes, and variable in different tissues. Did you say amplified?
Yeah.
I thought you were saying amphipathic. I'm sorry. I was like,
confused when you told her no, I told her.
I'm sorry. I thought you said amphipathic. But amplified, yes. Amplified response. Thank you for clarifying that for me. I didn't understand. Yeah, I was like, oh, okay. Okay. Sorry. No, I'm sorry. I did not hear you. So she was, Ms. Bannister was correct in that it does produce an amplified response, which is variable in various tissue types. Additional questions or clarification? In chapter 5, we summarize messengers, whether they be hydrophilic or lipophilic. We learned using those two words gives us at least 3 hours worth of dialogue. Okay? Just instead of saying it's water soluble or lipid soluble. We also had our world open a little bit and say that we get a lot of diversity in the target cells by using hydrophilic messengers. Lipophilic messengers tend to generally just called transcription, which isn't a simple process, but you start kind of see the diversity of cell responses. It's not as diverse compared to a hydrophilic messenger. We are going to look at different messengers, hydrophilic or lipophilic, all semester. Most notably, first at the nervous system, and then in Unit 3 we'll get into the endocrine system. And so what we're going to do, sorry, let me back this up, is for the rest of the second unit, look at how our nerve cells control, or I guess affect our target cells by releasing neurotransmitters. The bulk share of our neurotransmitters that we'll study are hydrophilic in nature. So we got to go back to understanding the target cell has to have a membrane-bound receptor and so on and so forth. And so in this nervous system, we're going to have considered this one-way talking, if you will. The nerve cell releases the neurotransmitter, the target cell binds to the neurotransmitter, and responds by opening or closing ion channels or activating a second messenger system, in essence. And we have a brief introduction to comparing that to the endocrine system on table 5.6 in your textbook. The nervous system, so real quick, before we look at any difference, these two systems are used to do what? To what? To control target cells. Releasing different chemical messengers, whether they be hydrophilic or lipophilic. We try to differentiate early in the semester that the type of control is more of a time difference, really. The nervous system can target cells very quickly, milliseconds. In comparison, the endocrine system is going to take longer than milliseconds, because we learned that some steroid hormones have to be first made, then secreted, and transported, and then infused to the target cell, initiate transcription and transfer, right? We're looking at hours, minimum. So the nervous system is going to give us a more quick, precise type of control by the release of neurotransmitter into what? Neurotransmitter is released into synapses. By contrast, endocrine system releases things called hormones into the bloodstream. What is common though, the targets still have a receptor to bring about a change in the target cell. What questions do you have over this kind of broad differences between the two systems?
What was the slide you had before that one?
Figure 520. Again, the brief comparisons in table 5.6 will elaborate for the next 3 weeks on the nervous system, and then 2 weeks after that on the endocrine system.